Yes. Yeah. Today we have six startup companies giving presentation later, and over 50 investors are invited. Most of the investors are from United States and China. The event is going to be live streamed on YouTube and Facebook as well. Today's conference is on site plus online. We have conference room in both San Jose and Hangzhou, China. Over 1,000 audience are online and on site. Thanks for coming. Today, my colleague Brian is going to have be another moderator taking over the on-site audience in San Jose. I heard that we have great food and wine at Cube San Jose for the party. Enjoy that, everyone. Before everything, we have a brief video for an introduction to take us to Cube together. Dan, please let me know if the video is ready.
Yeah.
Yeah, Brian, if the video is ready, you can go ahead and play that.
Yeah, we'll roll it.
Yeah. We cannot hear the song.
Our business model offers office workspace, life space, technology cooperation, business services, and startup incubation. Our goal is to be an all-inclusive incubator that offers every startup the opportunity to launch, scale, and sustain their business on a local and global level.
Silicon Valley is bubbling with innovation. At Cube, our goal is to create an environment where ideas, people, and companies can thrive. We look to provide the best resources and network to support our tenants and members' vision.
So far, we've attracted more than 30 companies to build up their business at Cube and helped more than 100 startups to grow up in our community. Companies here not only have access to our world-class facility, We also provide an accelerator program, the resources and events to help them scale efficiently and effectively in the global market.
Over the last few months, many of the companies at Cubase Center have secured tens and millions of dollars in various funding. Over the next few years, we look to position and equip our companies in new and exciting ways.
On behalf of Cubase Center, thank you for joining us today.
And now, lean in and get ready to hear from exciting new companies of the future. Welcome to Cubase Global Pitch Day.
Thanks, Brian. Thanks to the San Jose site. Besides the pitching company, today we also have 3 mentor guests for the comments. We have plenty of experience in industry and biotech investment. We need to hang on a minute. Yep, that's fine. Let's today we have 3 mentor guests and comment and for the comments we have plenty of experience in industry and biotech investment. We have Wu Chen Zhang from CRCM Venture. We have Tania Fernandez, partners of Dreamcatcher Ventures. We have Jiawen Han, Vice President at King Tour Pharmaceutical. The advice and comments will be very helpful and informative for the startups. Okay, let's get started. The first company, it's Vitanova Biomedical. They're from Texas. Vitanova is early stage biotech company focusing on delivering transformative cancer therapy with the use of light-activated intracellular acidosis. The company aims to improve the lives of cancer patients and the professionals who care for them by providing cost-effective cancer therapy that eliminates side effects. Today, we have Tom Roberts, the president and CEO, giving a presentation about it. Tom, you here?
Michael, can you hear me?
Yeah, we can hear you. You can go ahead and share the screen.
I apologize, but I keep hitting start video and it says the host
hang on a minute.
Has stopped my video.
Hang on a minute. You're good to go.
Okay. Are we good, Michael?
That's good. Go ahead.
Okay. Good evening, everyone. As Michael introduced me, my name is Tom Roberts. I'm the CEO of Vitanova Biomedical. We're an early stage biotech company with headquarters in San Antonio, Texas. And our mission is to develop and provide an efficacious cancer therapy which eliminates the side effects of current cancer therapies in the marketplace. I'm sure we're all familiar with the three main methods of treating cancer, the standard of care: cut, burn, and poison, or surgery, radiation therapy, and chemotherapy. All of these techniques were developed years and years ago, the earliest being introduced 75 years ago. They're quite archaic at this time. And in the case of radiation therapy and chemotherapy, an individual can be subjected to between 30 and 40 different dosages or treatments for anywhere between 3 and 6 months, quite extensive. Over the years, both private and industry have spent billions and billions of dollars on curing cancer, and yet still oncologists today still fall back on the standard of care of cut, burn, and poison, and hope. I'm sure we're also all familiar with the many side effects that these therapies cause. And there's too many to go through them all. But even the more recent therapies, such as hormone therapy, immunotherapy, and targeted cancer therapies, either have side effects or they're too early to understand what the side effects are. Prostate cancer is a little different in that you still have the standard of care of surgery, radiation, chemotherapy, which still have the side effects. But you also have a therapy called active surveillance. Prostate cancer is the only cancer where one of the therapies is wait, watch, and do nothing except monitor the cancer. And out of all the millions of men who have prostate cancer, 40% of them are either recommended or choose active surveillance. The issue is active surveillance also has issues. First of all, as you're monitoring your prostate cancer and not doing any other therapy, you have to have repeat, very painful biopsies. In addition to that, 30% of men discontinue active surveillance because they can't live with the stress of living with cancer and not doing anything about it. And then 60% of the men that are on active surveillance will actually progress to a more advanced stage of cancer later on. There is a clear and significant need for an effective prostate cancer therapy that takes the place of active surveillance and doesn't cause unwanted side effects. At Vidanova Biomedical, we've actually achieved this. We have developed a targeted two-step combination therapy. And by combination therapy, I mean it has two components. The first component is on the left top of your screen. That's a drug, and our drug is a nanoparticle-based targeted drug. The drug is injected into the tumor, and then the second part of our combination therapy is the drug is activated by a very deep penetrating laser light. And we have a partnership with a large medical device company that will supply our FDA-cleared lasers. For prostate cancer, it's a very simple process. The drug is actually injected intratumorally, going through the rectal wall. And then the probe to deliver the light is also delivered through the rectal wall. So there's no incisions, there's no cutting, and there are no side effects. So what's the mechanism that kills cancer. How are we achieving this? Basically, what we do is we create a very natural cell death process that already goes on in the body. We disrupt the regulation of pH inside a cell. And the way we do that is through our drug. If you look at this depiction on the screen, the green ball on the left is a nanoparticle. And the only purpose of that nanoparticle is to absorb very deep penetrating near infrared light that can go roughly 3 to 5 centimeters through tissue. And it actually flashes like a lightning bug. And it flashes the UV light that is necessary to activate our drug. Now that drug is depicted by the little blue hexagon on the screen. And what we do is through a chemical process, we attach that active ingredient to our nanoparticle. When we hit the nanoparticle with deep penetrating light, it flashes UV light and it causes our active ingredient to shed a hydrogen ion. That causes the inside of the cell, a cancer cell, to become very acidic very rapidly. It can't recover, and it actually goes through a natural cell death process. It kills itself. We deliver our pH disrupting compound directly to the cell through targeting. It's a natural cell death. There are no poisons. There is no burning. And only the drug that is exposed to the light is activated. If it's not exposed to the light, it actually passes the system. Now, we've tested this with multiple types of cancers in the laboratory. This testing was done with our key active ingredient, not with our nanoparticle, but we tested our mechanism on 2 types of prostate, 2 types of breast, and pancreatic cancer. And in all cases, once our drug was exposed to the light, it killed 80 to 100% of the cancer cells within 2 hours after light activation. Then we actually took that to a mouse model utilizing triple negative breast cancer, and we went through the same process. The tumor in the mouse was injected with our drug. We shed 2 minutes of light on the tumor, and you can see the mouse on the left was not treated by our drug. That was a control mouse. The mouse on the right that had a tumor roughly the same size was treated 1 time with our drug with 2 minutes of light. And we received greater than a 50% reduction in tumor volume. In addition to prostate cancer, this is a platform technology. Our core compound, that nanoparticle with our active ingredient attached to it, which is depicted in the left bottom of your screen, can be targeted to any type of cancer. You can see that at the top left, our first indication is for prostate cancer. And we've attached a prostate antibody to our drug, so it will only seek out prostate cancer cells and won't go into any other type of cells. Our second drug that we have planned is actually in the animal health space, and we are in negotiations with a large biotech company as we speak, and they have requested that we develop our drug for 5 different types of animal cancers, including melanoma and soft tissue carcinoma. We target that with a different type of antibody. And what's unique is our third drug using that same antibody for the human market will actually target breast, colon, and lung cancer. So we have a platform technology here. 90% of the drug for all of those cancers is already developed. All we have to do is retarget it. Prostate cancer alone is a $1 billion market. There are 3.1 million men just in the United States living with prostate cancer. 250,000 newly diagnosed a year, and 35,000 men are dying every year of prostate cancer. We project using the same cost structure as surgery or radiation therapy, that at our peak year, 8 years after launch, this could be over a $3 billion product, and that's with a 25% penetration. Our intellectual property is strong. We filed our IP in 7 different countries and or regions, and we have it granted so far in the European Union, the US, Mexico, Japan, and Australia. We're still waiting for China, and we're still waiting for Canada, but we're very hopeful and believe that'll happen. We have a fully owned license agreement from the University of Texas, and they are shareholders in the company. Our regulatory strategy is already mapped out. It's very clear. We will submit this as a new drug application to the FDA first in the US. And within that NDA application, we will also submit the second part of our combination therapy, the laser, with our drug. However, it's of note that the laser that we'll be utilizing has already been cleared by the FDA and is commercialized for other indications. We just have to take it to the FDA with our drug. We have a very seasoned team. I won't go through the criteria or the credentials for everyone, but myself, I was with Hoffman LaRoche for over 20 years. I ran product development for 5 years. I was a business unit head for the country of Canada for a $100 million business unit. I've developed and launched over 10 healthcare products globally. Dr. Godovan, who is on the line with us today, Dr. Godovan was a full professor at the University of Texas. He's a Dartmouth grad, and he is the inventor of our technology, the founder of the company, and my chief science officer. We also have a CFO who is not full-time right now. And then we have advisors to the company, both business and clinical. Our business advisors have a lot of biotech and pharma background, also startups and IPOs, such as Dr. Stock. We have 2 physicians, Dr. Peoples and Dr. Michael Liss, who are both affiliated with MD Anderson and also the Mays Cancer Clinic. So we also have the expertise from a clinical standpoint that we need from an advisory point. Right now, we are raising a bridge loan between $500,000 and $1 million for this bridge loan. We have already had a friends and family round that was very successful, where we raised over $1 million to develop our drug. We were also awarded a SBIR grant from the National Foundation for Cancer Research. This $500,000 to $1 million will help us conduct our proof of principle testing in an animal model with our completed drug and our final laser. And it will also allow us to develop the drug for our animal health partner, who, with positive results from this proof of principle testing, have committed to a $5 to $7 million investment to facilitate our drug through commercialization in the animal health space. And this is my last slide. I just want everyone to see that we truly have a very full and intricate plan from a preclinical, clinical, regulatory, and funding all the way through commercialization. We have this mapped out. We have all of our strategic partners in place. Everyone to help us with our investigational new drug testing all the way through commercialization. So today, the only thing that's slowing us down is a successful raise of the 500,000 to $1 million in bridge loan. And that is it, sir.
Thanks, Tom. That's a really nice presentation.
Thank you, sir.
Beautiful work. Are there any questions from the audience? You can just raise your hand or unmute yourself. I can. Minute, we have a questioning here. Oh, whoever is in the panelist, you can just go ahead and unmute yourself and ask directly.
Hi, this is Kevin Henderson. Can I ask a question?
Go
Sure,
ahead.
Kevin, please do.
You said on your tumor response, you can get an 80% kill rate in about 2 hours, and the cells undergo sort of natural cell death, the apoptosis. My question is, in patients, for example, who have leukemia and they have a fairly high tumor burden or leukemic burden, and they undergo rapid cell lysis, they develop something called tumor lysis syndrome, which is relatively, can be life-threatening. So my question and my concern is if you get an 80% kill rate within 2 hours and you have a fairly large tumor burden, my concern that the patient may develop sort of an anaphylactic response, and particularly if you're kind of having an acidotic intracellular environment that you're releasing into the systemic circulation, that can cause patients to go into shock. So what are your thoughts, or have you seen that? And then one question too, it seems like this would be more effective in localized therapy because the tumor would be localized and you would apply it there. How would it be how would it perform in metastatic cancer? Would you sort of apply the light therapy to the entire body, and how's that done?
Yeah, if I may, so Kevin.
Can you hear me?
Yeah, that's my phone a friend. That's Dr. Godovan, who when he believes I don't know the answer, he jumps in. So,
Kevin, I just
go ahead, Matt. Matt,
can you still hear me?
now we can.
Okay. Yeah. So when the cell goes into its apoptosis, it will express a marker for macrophages to come and destroy it. And we monitored the pH using optical dyes in the Petri dish studies. And we didn't see a significant increase in the extracellular pH. We're triggering the apoptosis with just a really rapid and profound intracellular acidosis. And once that's triggered, the cell, as it goes through its well-known apoptosis pathway, macrophages come and destroy it. I agree that right now the technology is perfectly suited for local disease, and we are looking into strategies to enhance the technology for metastatic cancer. And for example, the intratumoral injection is ideal for those types of tumors that are easy to access with an injection and the light source. But the drug itself could be intravenously injected and still make its way to the prostate tumor bed. We just know that we would probably be much more effective and efficient with an intratumoral injection.
Yeah, thanks. And Kevin, to Matt's point and to your question, our first indication is for local stage prostate cancer. So we are looking at least with our initial indication, a single tumor, localized.
Yeah. Hi, Matt. Just a quick follow-up question to that. What kind of apoptosis that goes to? Is a general apoptosis or necroptosis? Do you have any markers for that?
Yeah, we see apoptosis and secondary necrosis because the rate of apoptosis is so fast, and we've actually made videos of the cells bursting open. So it's known that with rapid apoptosis, you can get secondary necrosis.
Gotcha.
And we tested that early on with biomarkers.
I believe Tania got a question.
Hi,
Tania,
thanks, Michael. This is Tanya. I'm going to go where Kevin went. I think I share a similar concern, but pushing that a little bit more. Could you please explain the hypothesized basis of selectivity and specificity of the cancer cell versus the normal cell? So you're injecting tumorally into the rectal wall and disrupting the pH. Are you counting on the acidic environment in the cancer cell versus the normal cell to lend you that kind of therapeutic window? Is my first question. And then my second one, could you talk a little bit about the profile of the patients? Because Tom, I agree with you. There's 40% of patients are under surveillance, but wait and watch strategy is not going anywhere. And I'm assuming that's not your target population. You're targeting the ones that progress, the 60%. Yes. Could you comment on both, please?
I'll take the first question, Tanya, and the specificity is derived from two things. One, the nanoparticle is fabricated at a size that around 120 nanometers. It's too large to passively diffuse into any cell. The only way that it can go in is through a targeted endocytosis pathway. So we attach an antibody to the surface of the nanoparticle. In the first indication, it's prostate-specific membrane antibody. And we have excellent data that shows that that is bound and incorporated and pulled into the cell. So healthy tissue immediately adjacent to the tumor would not receive any of the drug. The second level of specificity comes from the fact that the clinician can very discreetly shine the light on the tumor and the appropriate margin that he or she is hoping to attain. And I'll let...
Got you.
Okay, and
thank you, Matthew.
To your second question, Tanya, actually, those 40% of men that are on active surveillance, we absolutely agree that active surveillance is not going anywhere, right?
Right.
Now, 30% of those men, 30% of the 40 actually seek a different type of therapy than active surveillance after 2 years because they can't live with the stress of living with cancer and not doing anything about it. So we believe that our initial entry into the market, our foothold into the market, will be with men that are on active surveillance that are actually seeking the next step therapy. And if they didn't have something like our therapy that isn't invasive and it doesn't cause side effects, they would be moving on to surgery. And I can speak to this because I am a man who's living with prostate cancer, have been through surgery and radiation and understand the regimes very well. So we believe our foothold will be with the active surveillance people who want to move on. And once it's been adopted and becomes more utilized, I believe it will spread, and some of those patients that are getting surgery who might not need it at that time because it's a little too aggressive, our therapy will bleed into that.
Thanks, Tom. Due to time, we gotta pass the floor to Brian in San Jose. Thanks, Tom. Really nice presentation, and thanks for coming today.
Thank you all for the opportunity.
Thank you. Brian, so for the second company, hi, Brian. You there? Brian? Hi, Brian. Are you able to let the second company get in?
Yes.
Yeah, the floor is
you
yours,
hear me?
Brian. Yeah, I can hear you. Floor is yours, Brian. Thanks.
Thanks, Brian. Hi, I'm Sikdar Zakir, founder and CEO of Histophy, and I'm a physician by profession and did clinical practice in internal medicine for 10 years. And in 2005, I defected to digital health. So since then, it's all my digital health life and lifestyle. So Histophy is a digital healthcare platform in cancer care. So it is headquartered in Los Angeles, but it's a Delaware C corporation. So what we do first is we capture and process data. So this is the foundation of utilization of artificial intelligence applications in cancer diagnostics, and then distribute insights real time to the physicians and researchers in cancer care. Ultimate goal is to optimize the treatment outcomes, which is very important in cancer therapeutic outcomes. So let me start with the problem. Number one issue in cancer care, be it in South Asia. I'm originally from Bangladesh, and so you will hear some reference to that country. So late diagnosis leads to a chain event, which leads to suboptimal treatment and uncoordinated care, which are the main culprits in the cancer care poor outcomes. So late diagnosis are due to two main factors. Number one factor is high cost of technology, and number two factor is the limited access to technological innovations and resources to the physicians in rest of the world. So what we do is, as I said, the number one factor is high cost of technology. So the physicians in the rest of the world, they shy away first until it is too late to adopt the technological innovations in cancer care. So we finance the platform for them. So whenever they are ready, we just shake hands, give them the tools, including a digital pathology scanner, which may cost up to 200,000. And then we make them partner, which are called Histophy partner providers in respective geographies. So they are our B2B clients where they do diagnostics using our licensed AI applications for cancer diagnosis in prostate, lung, or breast cancer. And then we move them to the treatment planning stage with further insights from the treatment planning process, like tumor board and instruments like that or tools like that. Then we submerge like a submarine underwater, because at the therapeutic stage we don't take part in anything. We only collect data. So we go to the B2C using a mobile app connected with a nurse navigator. So the nurse navigator help each and individual patients to navigate their journey to cure. So our platform is HIPAA and GDPR compliant, and all AI apps we used either CE, IVD approved for clinical AI apps or FDA cleared for research.
I think you're on mute. You probably click the mute. Hello? Hello?
converted the platform a digital one, very simple to work through. So number one, as I said, we finance the platform, including hardware, and equip them with AI-enhanced diagnostic and treatment planning capacity. The main target is to give them better insight, and at the second step, to connect them to the global network of oncologists, pathologists, subspecialists. I should mention here that 80% of the oncology subspecialists and pathologists in the whole world resides in the USA. In addition, in last 10 decades, 18% of pathologists shed away due to old age or retired and not being added. New ones are not coming to pathology. So at the third step with the mobile app, we do everything regarding treatment, follow-up, and post-treatment or post-cure monitoring of the patient at home. So it completes the entire ecosystem with our digital platform. I'm sorry, the slide is not moving. Oh. Yeah, it's not moving. You have to do manually. Oh, yes. Okay, this is in another way what patient does from step one, which is the symptom to point of care, then step two, point of care to diagnosis, then diagnosis to second opinion, and then the second opinion to treatment planning. All these steps are manual and physical processes. Either patient take flight or travel from one city to another, and it is a journey for 6 months to 1 year to 2 years, depending on the situation of the cancer. So just to give you an idea what patient does, and at the bottom you see our tools, which are constantly being made available to physicians and their patients. So who are these cancer patients? At the top, the red colored patient population, we have select 25 countries in Asia, Africa, and Latin America. And I've been in oncology practice over telemedicine for last 2 decades. So we found 8% of this patient population, they are like the super rich, or kind of they use Tesla. And the 47% blue colored population, patient population, they use hybrid mechanism. Sometimes they take flight, sometimes they travel from one city to another, sometimes they get high cost treatment at home. And the bottom 45%, they are like the used car user. So they don't have much flexibility. Whatever they find, they use until death. So what we do is through capturing of the data, that means digitization is the first or primer to all the activities we do. So that's what we try to keep very low cost, $15 per patient when they onboard into our digital platform, or our partner bring in one patient into the platform. The cost is only $15, which sets into motion subsequent diagnostic, therapeutic, at-home services. And that adds an average revenue for us of $125 per patient. So our providers charges patient and pays Histophy. So patient pay out of pocket to providers. In our marketplace, this is a cash market. 95% cost of cancer care is out of pocket. Only 5% is paid by insurance. So what does it do at the end of the day? There is 2 factors. Time is of essence in cancer diagnosis and treatment. So our turnaround time is 70% faster than the conventional one. So where it takes 40+ days for diagnosis and confirmation of a cancer, it takes only 12 days, even if the subspecialist is out of Mayo Clinic or Johns Hopkins. And this time, faster turnaround also reduces cost, which is 50%, a significant saving. So if I go back to that slide, the bottom population have the now option to move up to the blue population category or hybrid category. And the more hybrid category can move up to the top 8% category. So our market size is in 25 countries, and the total market by dollar is $3 billion care coordination market. And there are 8 million patients, as I showed in the color-coded patient population. Each of them is spending $375 today, whether it is in Bangladesh or Ghana or Sierra Leone. So our target or SAM is $125 million at year 2024, based on we are targeting to serve 1 million cancer patients every year from year 2024. So in our market population, we have targeted 300 HPPs or Histophy partner providers, mostly in low and middle income countries. And they are our point-to-point contact or our peers or friends from our more than 2 decades of digital healthcare engagement. So the competition is siloed, like in digital healthcare or AI-driven healthcare, there are competitors. But what they say to our clients is, if you want to use our AI-driven diagnostic tools, you have to, that is 10% of the ecosystem. If you compare it as a hexagonal relay racetrack, so they only provide 10% of the ecosystem, but charges $120,000 minimum per year license fee, whereas we charge $0, but they only pay per patient unit or unit-based payment to Histophy. So, but what we do, we completed the entire digital healthcare ecosystem for cancer patients. And we only ask our partners to share their credentials of the oncologist, pathologist. If we are satisfied, we shake hands at 50% of the milestone. So it didn't start, in a year or two. The architects behind, Dr. Jay Sanders is the founder of American Telemedicine Association. He's also considered, father of telemedicine. So we have been working together in telemedicine for last 25 years. He's now chairman of our advisory board. And Nizar Mamdani, he and I expanded the University of Nebraska Medical Center cancer programs using telemedicine, using my software, to 25 countries in 10 years until 2010. Nizar recently retired, so we developed these connections globally with all the oncologists and hospitals, labs. So that is what Nizar is bringing. More importantly, I had an experience with a patient who, suffered from lung cancer and died miserably. So I had taken help from Nizar. I didn't meet him before. In 2001, for the patient, I did the first telepathology second opinion consultation in 2001 between Bangladesh and Nebraska. And unfortunately, it was too late. And the patient died, and the patient was my own father. And then as a courtesy, after 5 days of my father's death, I traveled to Omaha to meet with Nizar because I found him online at that time. So I met him. I visited his wife in ICU who was dying of lymphoma. And she died just 1 month after I visited her. So this is how we are connected. And we architected the whole entire system. And the co-founders includes, besides me, 2 other women. On left is Associate Professor Laura Ike. She's a US Navy veteran, head of our pathologist. And on the right is Associate Professor Afsana Laizu. She's the co-founder as well and director of anatomy pathology. And she's my wife of 2 daughters. So besides the founding team, we have world-class people working here. Jeffrey, based in London, He is the IT guru and a business development guy. And Barbara Levy is the former vice president of American College of Gynecologists and Obstetrics. And she recently retired and joined as part-time. Ranjit Shetty is the software engineer with 80 startups and based in Austin. He is the CTO. And Anthony Lombardo is our part-time finance guy. So we are now seed fundraising. It's $2 million seed fundraising going on with another projected $3 million late seed funding. So we are right now at the box, rectangular box dotted with the blue dotted lines. And here we are taking these MVP, which are already in use, and shipping the whole system to 4 clients in Africa and Asia, including my native country, Bangladesh. Launching this MVP in January, finalize the product market fit, and we have to do some more mobile custom fit with language versions as well. Ultimate target is to create a repeatable customer acquisition strategy. Our each Histophy partner provider also serves other smaller hospital labs in their own geography. So we have some works to do. This is just a breakdown of what we are doing and what is the funding stage. We have used our own funding, friends and family, up to $1 million in last 2 years. And this is our requirement for next 2 years. So thank you for your kind attention. And what we are sitting in the hub of the technologies in West Coast. So this is where innovations are happening on daily basis in cancer care, in therapeutics, in diagnostics. But I have experienced when I was practicing in Bangladesh, and it took about 10 years time for any technological innovations to reach the end user, which are the patients in different rest of the world. So now our target is to make any innovation, intervention, and breakthrough available to rest of the world next month when it happens here. So I hope you'll find it interesting to expand the technological benefits to rest of the world and make some prudent investment into Histophy. Thank you very much.
Questions for you. Michael, are you there?
Yeah, thank you. That's really.
We have a couple questions in San Jose for Zach. So Zach, stay up here. Did you have a question? Okay, come on up.
Hi, Assalamu alaikum. Nice to meet you, Dr. Zakir. My question was in the solution that you offer the hospitals. I like the business model, and I think it was yourself who mentioned about this kind of innovation in business model. What equipment do you provide as part of the package?
Yes, our main target is to capture data, and in radiology, it has been digitized long ago, at least 10. Pathology was the last segment in cancer diagnostics, which is left behind. So now it is catching up. So we made sure that all our partner providers have the tools. It includes a digital pathology slide scanner, where you put in the slides, glass slides, and it scans, and then we can apply AI, tools.
So basically, it's already images from MRI, X-rays, and so on. You scan them, and then it's your AI software that analyzes the images together with the patient's data, and I imagine, general research data, and then comes up with suggestions and potential therapies and the triage process.
Exactly, and this is a very good question. And whatever data there are available, the AI application industry, especially in digital pathology diagnostics, they invested in last 2 years more than $500 million. So what can I do with $5 million? So we license those applications. So through different partners from leaders in the United States and European countries. So this is a marketplace for any innovations in cancer care to sell their products to end user. So that's how we license some part of it. We did not go into, AI application development. Having said so, the next step in our goal set is to create digital biomarkers. So that's what we are preparing ourselves for. Thank you.
Michael, are you with us?
Thanks. Due to time, we probably need to go to the next company. And thanks, Zach, for coming here. It's a really nice presentation. Brian, you want to give an introduction for the next company, or I can do that?
Yes, Michael, can you give it while I'm setting up the computer?
Sure. So for the next company, we have Boris Sorcha, and it's going to be on-site presentation. A little bit introduction. So Biosocial is it's a targeting on microbes, so has unique access to the hidden chemistry of the microbiomes. The only capability in the world to turn the long sought but previously unoptimal drug-like molecules directly from microbes, leading to the direct access of the hidden molecules and the filing the key missing data. Today, Ross Youngs is going to talk about it. He's the founder and CEO of the company. Ross, you can go ahead.
Terrific. Thank you so much for having me today. I appreciate it. So what we're doing is we're going to build a valuable pipeline of immunomodulating opportunities with partners and collaborators. How do we do this? We're going to unlock nature's microbiomes for therapeutics. So where is the problem itself? One of the things that we have learned in science is there are tens of thousands of metabolites from microbes in the human. Specifically, a lot of them reside right in the gut microbiome. These are virtually inaccessible. They're in such small quantities, you can't just go get them and understand what they are. In fact, less than 140, about 140 are known and reside in the human metabolome database out of tens of thousands of microbial molecules. The interesting aspect is the microbiome has now been associated with virtually every disease state, and it's impacting every aspect of our life. The vast majority of the small molecules that circulate within the human system, within our own blood, are actually from the microbes themselves. So virtually all technologies have focused at getting at these hidden molecules indirectly. Indirectly does not tell you what these molecules are, and most likely they never will because it's such a complex system. So what we've been able to do is we have invented, scaled, and provide access to this chemistry from natural microbiomes, making available the most sought after drug-like molecules for researchers to discover new therapeutics and other products. What we know is that microbiomes around the planet have overlap. And in fact, this overlap is pretty dramatic because all microbiomes evolved from basically the same life. We've been funded in our technological pursuit by ARPA, DARPA, and the US Air Force by more than $9 million. We're completely peer-reviewed and proven. We're also done with research and development for collecting microbes at scale at the quantity and quality needed. So one of the overlaps that's critically important is the Terra Ocean project proved that the overlap between the human gut microbiome and the ocean is more than 73% genes and metabolic pathways. So if you can't understand what's going on in the human gut directly, we can use other microbiomes as surrogates. And when you think about it, most of the microbes that live inside and on the human were environmentally sourced in the first place. Now, where do the metabolic pathways get turned on? They get turned on in microbiomes. They don't get turned on in culture plates. So the reality of it is going directly to microbiomes to do the recoveries is the way to do it. But the hurdles have been having enough quantity and quality. Part of this is achieved by our harvesting technology itself. Part of this is achieved by having a deconvolution facility that can handle the massive amount of biomass that's needed to understand molecules that reside in microbiomes at nano and picomolar quantities. So let's talk about scale a little bit. If you're talking about unculturable microbes from the natural environment, a liter sample is pretty much about what science has been able to study. I found one study at UCSD where they processed 1,000 liters, and this wasn't to get the microbes. This was to look at the genomics. We are at a whole different scale. And the scale is what allows a deep dive into these hidden molecules. So our scout vehicle, which is proven and can scout a microbiome in virtually any habitat, processes a minimum of 10,000 liters. Our full-scale harvesting unit processes 20 million liters and delivers 1 million grams dry weight of a microbiome representing 500 to 1,000 species, and allows us direct access to a minimum of 70,000 small molecules made by microbes. So how do we end up making money? Why is this such a market opportunity? Well, the reality of it is cost of drugs, cost of discovery itself has skyrocketed over the last 40 years. Part of the advantage of microbial-based drug discovery is these molecules are already pre-optimized for biology, not only for therapeutics, but quite frankly, for agrochemical, for cosmetics, for sustainability. This could be a life science revolution in the making. Our goal is also to sell rare molecules. What we've seen in our harvest is we've seen molecules that don't reside in libraries today, but are wanted. So when you go to the different library companies, Sigma, Aldrich, and other people, and you try to find a molecule, it may not be there. And if it is, it can cost an enormous amount of money. What we've seen is access to those kind of molecules in very large quantities. We also know that with our ability to interrogate the chemistry and then test that chemistry in assays, because the quantities we have are enough to test in assays, that we bridge the data gaps for AI and computational. When you have the capability to understand the genomics, the metagenomics, the metabolic pathways that exist within a microbiome, but on top of that, understand the small molecules and understand the activity of the small molecules, the gaps in data sets will be bridged rapidly. So where else does this play, and what kind of products can come from this? There is such a wide range of opportunity. That's why Biosortia is first starting in immunomodulation, because that opportunity covers quite a few disease areas, not only cancer, but infectious disease. It covers autoimmune, and it covers untreatable asthma. There's just dozens and dozens of disease states that can benefit from us uncovering molecules that modulate the immune system. We've had the opportunity to work with a number of people, not just partners, collaborators, and key vendors. One of the most important things about what we're doing, and that is technology has caught up with deconvolution scale. And we can thank the cannabis industry for this, because in the cannabis industry, they've been wanting to get at all the small molecules that reside within cannabis, including all those cannabinoids. In our particular case, the extraction technologies have advanced dramatically. That accelerates utilizing high quality recoveries of microbiomes and their deconvolution. So these can be plated onto well plates and tested by researchers all over. We have demonstrated this. We have a couple scientific papers that have been published. One of them was actually received the American Chemical Society's Editor's Choice Award, but we've worked with Lilly, the James Cancer Institute, Leidos, University of Rhode Island, and others in finding hits at subscale that were novel small molecules hitting targets. When we go to scale, our ability to be able to find these materials rapidly and make these available to researchers goes up dramatically. We have put $21 million into it at this point, with that $9 million coming from non-dilute. I've put in $4 million of my own money to drive this company forward, $2.4 million from board members. We're a J-Labs graduate and we have multiple patents issued, but as we move forward, we will be a patent powerhouse, creating potentially hundreds of patents annually. We have a high quality board, advisors, and team. We've got people that have been head of strategy for Gilead that's on our board. Dr. John Riles himself has started 3 microbiome-based companies, including AgBiome and Metabolome. What we plan to do is we're raising $20 million right now. We have a lead that comes from a top 3 tech firm, big tech firm. We will execute the microbiome factory at scale. That is technically the deconvolution facility that can handle the biomass. That would be a facility that would rival any of the cannabis deconvolution facilities that exist, but have the technology to plate out tens of thousands of small molecules. All of that is off the shelf. We don't have to invent a thing. We also will be executing the data bridging between genomics, the chemistry, and the activity. So we're going to put together computational facility and AI facility so that we can take advantage of what we know about these materials and how we can prioritize the wells for certain disease states. Be happy to talk about with anybody individually after this presentation or in the future. So what we'll be doing is we will be driving revenue at the end of our very first year of execution. We'll grow rapidly to over $75 million within 3 years of revenue. Collaborations driving IP and building a valuable pipeline, because our model is not to be the pharma company. It's to be the discovery company. We will be working with researchers worldwide with their assays, their screens, their targets to bring them the hits that they need. All of that will occur under licensing agreements, upfront payments, ultimately when it reaches that stage, milestones and royalties. Additional growth revenue from that data licensing I've already talking about, and that known library. We believe exits by IPO, acquisition, or asset sales. An example of that is as we put together a research deal with a major pharma, particularly in space around neuro-CNS or immunomodulation, we believe ultimately they'll want to buy that pipeline that's in existence, and that's an exit. I want to thank everyone. This was a short presentation and sweet. Hopefully there'll be some questions. And again, I'd like to thank the sponsors, Cubase, for having me here today. So thank you.
Thanks, Ross.
Michael, we have an in-house question, and then I'll pass it back to you to field some questions.
Well, first of all, Ross, really, really impressed. Huge congratulations to you and your team. Just astounding of what you've been able to do so far and how much skin and passion you have in the game. This is absolutely remarkable. I was really taken away by the number of partnerships you've had. One name and company really stuck out, and that was Illumina. So I'm sure you know Dave Walt very well, one of the co-founders, and I was actually on a call with him, 1 or 2 weeks ago. So a couple of us over here, Tan, myself, Andreas, we're part of this brand new initiative from the VIS Institute at Harvard, and Dave Walt is one of our advisors.
Fantastic.
So I'm curious, so in terms of number of partnerships you have, how do you, you have a lot of moving pieces, a lot of different segmentations. How do you leverage some of your partnerships like Illumina and others? Then I have another 1 or 2 questions after that.
No, that's terrific. Leverage is key in business. And you've got to be able to put the teams together and be able to put the partnerships together in order to affect change. I've always been big on creating high-performing teams, fearless teams that address the issues, that create accountability and get things done. I think when you get a lot of very smart people together and you give them the tools to execute, you will achieve. And we've seen that time and time again in every business I've been involved with. So it's a matter of structure. In the case of what we're going to be doing on genomics, metagenomics, and ultimately shotgun genomics to really explore microbiomes, I can't do that. My internal team can't do that. We're going to do a lot of leverage to make sure we're professional at that.
Great. When it comes to AI and leveraging partnerships with AI, so over here, Andreas, he's a senior machine learning engineer for my company, and he's also doing a lot of fascinating things over at Stanford Sleep Lab and one of the finest machine learning engineers in the Valley. A lot of his work is published in NIH's National Library of Medicine. So would you be open to partnerships with, say, our company, which is an AI powerhouse?
Absolutely. We see so many opportunities to take advantage of this data, not only when the data sets are filling up and complete, but even prior to that. We get substantial mass spec data. And one of the things you see pharma companies doing and research companies doing, they're mining the existing molecules for other opportunities. And they can do that through AI by looking at a set of molecules and saying, this is a learning set. In our particular case, we've got this wealth, tens and thousands from one harvest, molecules humanity has never seen. The best way we can prioritize is compare molecular structures, even mass spec data, to what we have within our libraries, because mass spec data is easy to collect. Structural data isn't. We've already seen that working, but we need to step it up a notch and take it to a whole different level. So our ability to find the right teams and work with them, we're excited about that.
Excellent. Exciting times for sure. We'll definitely stay in touch. I'm actually flying into United Kingdom next week. I'm sitting down for a 2-day symposium at Oxford University, and they also have strong interest in AI and genomics with our company. But that said, I want to pass this off to Tan. I feel like he has a couple of questions potentially.
One of the things I will say, there was a scientific paper published in January of this year talking about AI and its real possibilities. And it really highlighted extensively the fact that the data is not there. So one of the things in order to really make AI useful in every approach is to fill that missing data up. This kind of technology can rapidly fill that data.
Excellent. Thank you so much, Ross.
Hi, Ross. Great presentation. And just like what my friend, what Tony said, is that there's nothing like a CEO with his own skin in the game. And your skin in the game is, compared to most of the Valley, it's quite deep, I would say.
It feels pretty deep, but one of the things I'll say, and I thank you for that, is I've always been a platform kind of guy. And what I love about platforms, and this is in my mind, a brilliant platform. You can fail here or there, but you have so many opportunities. I never liked the true biotech model. No offense to the biotech model. The failure rate for a particular molecule is high. I'll be working with hundreds of those kind of molecules. I will have failures, but we will also have successes. So I love platforms, and that's one of the reasons my skin is in this game.
Me too. I'm exactly doing the same. Although I don't have as much in terms of quantity of those greenbacks as you have in your game, but certainly 3 years of time. The question I wanted to ask, there's one point I wasn't exactly clear, and perhaps you could clarify. So you're looking at the metabolites produced by your biome, and then creating profiles about those metabolites to see how they're affecting disease states, and selling that data to the pharma companies. Am I right? Or
that will be part of it. So that's not our main revenue driver, and it certainly won't drive revenue for the first several years.
Okay.
So AI in the first part of what we'll be doing will be basically understanding the data sets we need, utilizing the data sets that we have to set priorities. Ultimately, those data sets will have tremendous value. One of the things that happens with metabolic pathways within inside of microbes, a lot of them overlap. A lot of them have been horizontally or vertically impacting all other life forms. So these metabolic pathways, we're going to be able to point to the actual chemistry that results from them. And that's what's really important. A lot of the work that's been done in genomics to look at molecules shows precursor molecules. It doesn't show the complex molecules that exist in life. And this is the approach to get those complex molecules.
yes, I certainly see genomics, mRNA, gene expressions, then proteomics, and then next is metabolites. So,
Exactly. And with these microbiomes, the wealth of information that is there has been virtually untappable.
and one last, it may be a slightly controversial question, is what do you think of these companies at the moment that are selling consumers, send us your feces, stool sample, pay us $150 a month, and with that, we'll be able to help you lose weight, become fit, sexually more, strong, blah, blah, blah. What do you think? I personally think the science isn't there for those consumer services to be sold yet.
Well, I think you're correct. I think that it's a challenge, and it's going to take a lot more data to understand it. And the complexity that exists within any human's microbiome cannot be summed up with a quick overview. And I hate to put it that way. It is so complex. Now, I probably would prefer it if they weren't making diagnosis or suggestions. That, I think, is a very tough part with what knowledge we have around it. Can it help some people? Yes. Can it hurt other people? Maybe. Can people improve their own gut microbiome by following what science already knows? Absolutely. So I would say go for a diverse microbiome, go for one that is naturally obtained by the foods you eat, and eat healthy.
Ross, thank you.
Thank
In
you.
the interest of time, Michael, I'm going to pass it back to you. Ross, well done. Thank you for joining us. Great questions. Michael, are you there?
Thanks, Brian. Thanks. Yeah, thanks, Ross. Great presentation. So due to the time, we're going to cancel the coffee break. We're going to continue. The next company is going to be In Vivo Sciences. It's going to be online. And In Vivo Sciences provides phenotypic screening service for first-in-class drug discovery. The combination of the tissue engineer technology with assay automation and sample miniaturization offers a rapid and cost-effective platform for new therapeutic discoveries. Today, Ted Wakatsuki is with us, is CSO of the company. Ted, can you hear us?
Yes, can you hear me?
Yeah, you can go ahead and share your screen. Thanks.
Okay. Share screen. Am I sharing the right screen?
Yeah.
Okay. Thank you. So today I'd like to talk about our therapeutic program, IVS-201. This is for the precision therapy for the heart failure with a preserved ejection fraction. And this is the group for the oncology interests. So first, I'd like to start with the why heart failure is connected to the cancer. So this slide shows you the heart disease of the cancer patient. So standardized mortality ratio of the fatal heart disease among the cancer patient is quite high. The more than one means that the fatal heart disease is killing the cancer patient more than the patient doesn't have any cancer. And that suggested two things. The one is the cancer therapy by itself has the maybe toxic effect for the heart. And the second is the pathologically or the cause of the disease of the two diseases, heart diseases and the cancer are common. So this commonality is when you look at the literature, the recent paper publications, it's quite clear because comorbidity of the heart failure and the cancer is very similar, diabetes and other things, and also the cause of the disease, inflammatory stress, oxidative stress, is causing both diseases. So the pathway regulate these diseases, most likely the common one. So because of the reason, there's a new clinical field called cardio-oncology is emerging. So this emerging field, if you go to the large hospital and the academic institutions, there's a new group take care of these patients, cardio-oncology patients, and then there's many good training going on to treat this patient collaboratively with the oncologist and the cardiologist. When I switch to a little bit of a discovery issue, the cancer field has been advanced to develop the targeted therapy quite a bit compared to the heart failure drug development field. For example, the HER2 positive breast cancer, when you find this patient, there is a specific treatment for the HER2 cancer patient, but those type of diagnostic approach and the treatment approach for the heart failure is missing. So because of this slow advancement of the precision medicine platform, the FDA issues the new guideline. This guideline clarified FDA's position to support the drug development effort for the functional endpoint, even without much of favorable effect on the survival and the hospitalization. So that changed the landscape quite a bit. And one of the company that took the advantage very first time is the MyoKardia in San Francisco. And they developed the drug for the small population, but the targeted population of the hypertrophic cardiomyopathy. But this drug is proven to be improve their symptom. So they exactly took advantage. And this company is acquired by the Bristol Myers Squibb recently. So based on this cancer connection, I'd like to introduce our IVS 201 program. This IVS 201 is a small orally taken drug candidate and small molecule. It's categorized as an orphan drug to target specific type of heart failure within the heart failure with a preserved ejection called HFpEF. And this will relieve the stiffness of cardiofibrosis without affecting the physiological function of the heart. As I said, it takes the advantage of 505(b)(2) pathway so that, for example, this pathway has been taking advantage for the COVID drugs significantly, and this shows this pathway is very effective to bring the effective treatment to the market very quickly. So we are taking this pathway. And then this compound, IBS 201, is discovered by using our platform technology, which is made by the sample screening of the patient-derived heart muscle cells in vitro. They call new heart. And this platform is scalable. When you talk about the heart failure with the preserved ejection fraction, in general, the heart failure in the United States, about 6.5 million patients exist. And by 2030, this number will rise to the 11 million. And about the half of the heart failure patient is categorized as a HFpEF. Although the significant market size to treat the disease, there's not many effective treatment is available at this time. So this is a significant unmet need. There are two major problems in this HFpEF therapy development. One is the limited success for the one-size-fits-all drugs, because this patient of the HFpEF patient group are heterogeneous. So basically, they're categorized as a HFpEF, but there are many different type of syndromes is included in there. So it's very difficult to treat so many different type of patients. So recently, there are so many phase 3 clinical trial fails. They couldn't meet the endpoint. So that's a significant unmet need. And the second issue is the more biological one. Some type of HFpEF patient has the significant change in the structure of the heart muscle. The one is the cardiomyocyte within the heart muscle is enlarged, so they contract much more. And at the same time, the region of the fibrosis increase, so that the stiffness of the muscle is become so high, so that efficiency of a pumping function is reduced. So our solution is twofold. First one is to diagnose the HFpEF patient population and then classify different groups by using a traditional functional testing approach, using the echocardiogram or catheter pressure measurement and MRI, and using the AI approach to classify those patients. And then we can use those already existing data to train our AI platform. And also we piggyback with the biopsy sampling protocol approved for the another type of orphan heart failure program. This orphan heart failure is often misdiagnosed with the HFpEF. So this is a good sample group. And we expect to find the group of patients with this procedure that has a significantly elevated fibrosis and significantly elevated diastolic stiffness, so that our compound, which targets to the releasing the stiffness of the cardiac fibrosis, will be effective. So basically, this patient stratification select a patient that will work for the IBS 201. So our patent position, we filed the method of use patent for the IVS 201. In addition, we have another 15 class of compounds, also repurposed compound for treating a different type of heart failure. And we also have the right to use this the AI integrated precision medicine discovery platform. In addition, last 10 years, we have been filing many different method patents to screen this compound, making the platform technology to discover the precision medicine platform. So that's filed, and then also many of the patents are granted in a different region of the world. Our market opportunity is in terms of the HFpEF population in the United States and Europe and Japan combined is about 5 million. Many different reasons they may not be able to have access to the hospital and technology, so reduced to the 10% of the population. And then lastly, maybe about the few percent of the patient will be advancing to benefit our treatment. Because of this precision orphan drug treatment, by contacting the strategic pricing expert, it's 100,000 is a reasonable starting point. So we expecting our first market size is about the $16 billion. And this will be backed up with the previously mentioned acquisition of the myocardia by the BMS. So in terms of the competition, there are many companies, including the large pharmaceutical companies, discovering the heart failure treatment. But we chose two companies, very interesting companies, related to our situation. So one is a Cytokinetics. This is in San Francisco company, and a myocardia is also San Francisco company. But this both company target cardiomyocyte to treat the heart failure. And Cytokinetics chose a large clinical trial endpoint of a morbidity or mortality. But unfortunately, this phase 3 clinical trial didn't meet the significant endpoint. But the myocardia, because they focused on the functional endpoint and also targeted population, they were successfully complete the phase 3 clinical trial with around 250 patients. So we would take a similar pathway as a myocardia. We will use the stratification technology to focus on a specific type of heart failure. We focus on non-muscle cells, a new type of therapy to treat this patient group effectively. In our group, unfortunately, we couldn't, Ida Nach, our CEO, couldn't join the meeting, but she's leading the company for many years, and I am a CSO of this company. And I should mention the involvement of Dr. Yossi Schlesinger, who is the chair of the Yale Pharmaceutical Pharmacology Department. And you may know about him because he's a co-founder of many oncology drug company, including the Sugen. And recently we recruited the Michael Rich from the Washington University, who is an expert in the HFpEF medical field. So far, we have raised about $8.5 million through our founders' money, and also grants and contract, and then early revenues of the services that provide using our platform technology, NeuHeart. And we also have a different type of disease model with a NeuHeart for the not HFpEF, but the more genetically driven the heart failure models. And then some companies are interested in this technology. So we have a NDA with that company to negotiate how to develop this drug further with them. Our development plan for the next several years is following. And at this time, we think that the cost to finish the phase 2 clinical trial if we successfully enter the IND stage as the 505(b)(2) pathway, it would estimated to be costing about $12 million, and that's what we are raising. And we are also open to milestone-based trenching raising of a smaller amount to continue us moving forward while we are raising the large amount of funding moving forward. So to summarize our program, we have interesting drug discovery technology and effective to identify our anti-fibrotic drug for the precision medicine, IVS-201, using this platform technology. We continue to develop this clinical program of the IVS-201 to the next stage, but also we are making a collaboration with the large pharmaceutical companies and biotech companies to use our platform technology to develop another type of precision medicine for the heart failure. And thank you for the attention, and I'm happy to take any questions.
Thanks, Ted. It's a really nice presentation.
Thanks.
Are there any questions from the audience? If it's online, you can just unmute yourself. If it's on site, you just need to ask Brian for the mic. If you're an attendee, you have to raise a hand so I can give you the access.
Michael, we got a question.
Okay.
Hi, Ted. I
Yep.
just wonder for this molecule IVS-201, is this already FDA approved? Whether itself has a valid composition matter pattern or not?
Yes, it has a composition a matter pattern, but this patent is expired. And this was not approved in the United States, but approved in outside of the United States for the different indications. And therefore, once it's approved, it's a different, interesting situation for the FDA. We believe that this will be it's possible to go to the 505(b)(2) pathway because of there are another phase 2 clinical trial completed of this compound for the completely different the pathway. So that's the kind of profile of this IVS-201.
And that is developed by another company for a totally different
indication. Yes, exactly. Yes.
I see. Are you trying to change the PK/PD by reformulation?
No, we are following. So original approved formulation is different, but another phase 2 clinical trial used the oral formulation, and that was approved by the FDA to move on to the phase 2, and we follow that formulation. So we are not changing for the formulation for that.
Got it. Thank you.
Thanks.
I got a question from online. It's asking for the IVS-201. Is this small molecule, and do you know if the what is the target? Have you done any targeted convolution?
Yeah, so because of the repurposed compound, so the target is well characterized. We also did our own characterization, and there's an animal study, and then human data also exist.
Any stability get involved through this compound?
Safety and stability has been, basically proven by the clinical trial.
That's great. Thanks, Ted.
Thanks
for coming today. A really nice presentation.
Thank you.
So the next company is going to be on site. It's going to be Arise Precision Medicine. Arise Precision Medicine is a biotech company that developed drug candidates for cancer by breaking big problem into smaller and smaller problems. ARISE is trying to design a specific way to stop the cause of cancer, then target and protect the delivery system, and finally concentrate the drug inside the cancer cells, but leaving the normal cells undamaged. Brett is also an old friend. He is a founder and CEO of ARISE. He is going to be on site to give us a presentation. Brett, you here?
I'm here, yep. Are you seeing the slides okay?
It's great, it's great. Go ahead.
Wonderful, good. Thanks so much, Michael, and everyone for allowing us to participate in this conference. My name is Brad Niles. I'm the CEO of ARISE Precision Medicine, and our tagline is the PRDM oncogene targeting company, based on the focus on this family of genes and proteins known as the PRDMs. And the reason that we have focused on these is we believe the cure for cancer exists by targeting cancer at the genetic root cause of the cancer. Unfortunately, there's a major unmet need in many cancer types, but in specifically with lung cancer, the 5-year survival rates are exceptionally low. As you can see here, in cancers that are diagnosed at stage 4, below 10% 5-year survival. So clearly there's a major unmet need from the drugs that are currently existing in these later stage cancer patients, as well as the earlier stage. Up to 40% of lung cancers are diagnosed at stage 4, a very large population of lung cancer patients badly in need of new therapeutic options. The reason that we believe we can have a difference over the options that are currently available is based on a siRNA targeted approach that we're taking that differentiates ourselves from the other companies developing products in chemotherapy, immuno-oncology, and targeted inhibitors. These products, while they do kill some cancer cells, it's often in a small portion of the patients. A lot of harsh patient side effects, as we've been discussing a couple of the talks here tonight. We believe we can get around those issues through our specific approach, as well as having a precision medicine-based biomarker, because we are going after the genetic root cause of the cancer, allows us to identify those patients early, diagnose early, treat early. You have a much better chance of saving those patients. So the focus of our talk here tonight is on our lead candidate called ARISE-47 and its target PRDM2. PRDM2 is a part of a family of 16 different PRDMs, histone methyltransferases. It's been shown in a paper recently, the PRDM2 methylation disruption was observed in 73% of the tumors, as well as 20% of surrounding tissue as well. Clearly, there's a major impact going on in the lung in these patients with this disruption in PRDM2 and its isoform RIS1. RIS1 is a known tumor suppressor gene. Thousands of publications on this family of PRDMs, and specifically in PRDM2. And by use of this biomarker, as I was mentioning, we can select patients that in a clinical study that will allow for a faster study, a less expensive study, and faster to market. That's a big appeal to pharmaceutical partners that we would like to partner with for development of this therapeutic, making it a highly attractive candidate. So what are these PRDMs? It stands for the positive regulatory domain containing methyltransferases. And so they are the master regulators that sit atop a number of the key cell signaling pathways that have gone awry in cancer, things like c-Myc, p53, that are supposed to suppress cell differentiation, proliferation, and metastasis. However, there's a disruption that has occurred that results in a cancer by things like age, environment, and viruses that all lead to disruptions in PRDMs in a couple of different ways, including a silencing event, or alternatively an overexpression event, or this third option where we've focused our work is in the activation of a truncated form. So this is a smaller version, an unregulated version that exists in the cancer cell, but not in the normal healthy cells. So this gives us a great therapeutic target to go after where we can kill the cancer and not have an effect on normal healthy cells. So these PRDMs fit in a larger family of histomethyltransferases. As you can see, there's been a lot of work done in identifying this methyltransferase family. There's about 70 members altogether, and work has begun in the last decade or so on commercializing this family of methyltransferases. You can see some work done by well-known companies like Constellation, Epizyme, GSK, and as evidence for popularity of this family in recent years, you can see that Constellation was recently bought for 1.7 billion just this past June. So we're very excited to focus on this area, the PRDMs, which have not yet been commercialized. We are working to own the IP space covering the PRDMs, really carving that out as a niche for our company to go through and develop targets and therapeutics. Each one of the PRDMs has been very well studied and has been shown to be implicated in a number of different cancer types. As you can see here, over the course of the 16 different family members, we're hitting a lot of the major cancers by one disruption in one PRDM or another. So we've begun our work here, and it's obviously kind of a large potential platform, a kind of intimidating place to start. So we had to pick our places to begin our studies. You can see here in red some of the work that we've done in those cancer types, developing sRNAs against the particular PRDM. Each one of these red lettering denotes a cancer where we've observed more than 80% cell killing with our various different sRNA drug candidates. So we've narrowed down our product pipeline. We have a lead asset identified, PRDM2 for lung cancer. We also have assets that target ovarian cancer, breast cancer, multiple myeloma. In addition, we are working on a project for a small molecule approach as a targeted way of going back and altering these PRDMs and providing a good therapeutic option. So focusing on PRDM2 and its isoform RIS1, this is some data that is in the Human Protein Atlas. You can see here circled in red in the lower left. These are 5-year survival numbers in patients that either have high expressing RIS1 levels or low expressing RIS1 levels. So a big difference between the two, really implicating the role of RIS1 methylation in the pathogenesis of cancer. How about causality? This paper by our collaborator Shi Wang quite a few years back now, who first identified these PRDMs, found that in a RIS1 knockout mouse, one of the most common cancer types that developed spontaneously in this mouse was lung adenocarcinoma, really demonstrating a direct role between disruption in RIS1 and lung cancer. So we've taken the approach of how can we correct this methylation silencing event that occurs in lung cancer, the RIS1. And the way that we've gone about doing that is actually to identify the inhibitor of RIS1, which is RIS2, which is a truncated version of the PRDM2 isoform that lacks the regulatory domain. So by inhibiting the inhibitor of RIS1, we're actually able to increase the levels of RIS1 in the cancer cells. Tumor suppressor gene directly leads to cancer cell death. So a little bit of data demonstrating that concept. You can see here in blue, knockout of RIS2 with our asset ARISE47. And along with that, we get a more than 3-fold increase in the levels of RIS1, the tumor suppressor gene. So we're dramatically affecting this balance in between RIS2 and RIS1. And ultimately leading to cell death. This is measured in cell viability assay here in the A549 non-small cell lung cancer cell line. And you can see here, the second set of bars over with our therapeutic ARISE47, we're getting more than 90% cell killing with just a single administration of this sRNA to these cells. This compares very well to the results in normal cells, where ARISE47 is having very minimal effects. Demonstrating a great therapeutic index for killing cancer cells without killing normal healthy cells. Compares very well to some, the standard cisplatin chemotherapy approach, which kills both healthy cells and cancer cells at about the same rate. We do see some combination effects as well. You can see here with a combination with cisplatin, we're able to take those a few remaining cells that exist from our RISE-47 and really eliminate the total cancer cells with that combination therapy. So with some really exciting in vitro results, we were motivated to move forward into the in vivo animal mouse models of cancer. And so we partner with a company called Keystone Nano to help us deliver our siRNA systemically. This is an IV administration. This nanojacket technology is more than a decade in development, has been shown to be very safe in the body and has been shown to be very effective at aiding in the protection of the siRNA while it's in the bloodstream, as well as the uptake of the siRNA into the cell, released from the endosome, allowing the siRNA to do its mode of action inside the cell. And so by loading this, our RISE-47 in this nanojacket drug delivery system, you can see here in comparison to in red, the untreated control, we're getting more than a 50% decrease in tumor volume with this administration of the RISE-47 nanojacket combination. So we're very excited to have this as a proof of concept demonstrating PRDM2 as a druggable target in lung cancer. We're now going about optimizing delivery and dosing. We think we can have even more significant effects on efficacy of killing this tumor xenograft. But already we're meeting the threshold that exists for standard of care chemotherapeutics that are in use. Docetaxel here shows in the same model of similar 50% decrease. This is an FDA approved product, fully optimized dosing, and we're already reaching the level of that product. And we think we can do even better now with optimizations as mentioned. So where do we stand so far? We've identified our lead candidate. We've screened more than 100 siRNAs to identify our lead candidate. The results in an 80–90% cell killing. We've developed this concept of inhibit the inhibitor, demonstrating the mode of action for this siRNA approach. We have a selection of an in vivo drug delivery system through this partnership with Keystone Nano. We've been able to demonstrate very significant results in vivo in a mouse model of cancer. And as I'm mentioning, the next steps are to determine the fully optimized dosing and delivery, and then go into the pre-IND tox testing that would be necessary for an IND application with the FDA. Our goal is to license this off to a pharmaceutical partner. So we've already had a number of very promising conversations with potential partners, J&J, Pfizer, Lilly, AstraZeneca. Most any of the major pharmaceutical companies have an interest in products for lung cancer. So our goal over the next 2 years is to eventually get to the initiation of clinical trials. So we'll do that by optimizing our efficacy data, having a pre-IND meeting with the FDA to go over IND enabling tox studies, carry out those studies, file the application, and initiate the trials with a pharmaceutical partner. This provides an exit opportunity as well as many deals with pharmaceutical partners are taking place at this stage. We have a great intellectual property position in place and growing. We already have 2 PCT applications out there covering PRDM2, PRDM14 siRNAs. We also have 3 issued patents covering PRDM3 and various drug delivery systems for that. We have an exclusive license with Keystone Nano for delivery of the siRNAs with the PRDM-based siRNAs. This is covered by a 15 different patent family. We also have additional applications covering other PRDMs within the family as we try to really own the whole entire space of these PRDMs. This gives you a visual representation of these various different categories of the siRNAs, a very large patent family developing. As I was mentioning, our goal is to license off to a pharmaceutical partner. You can see here that about half the deals are taking place at the preclinical phase, and then adding another 15% in at phase 1. So a lot of the deals are happening at this early stage, with fees that are between $20 and $50 million upfront, that also have multiple milestone-based payments as the drug candidate advances through multiple stages of phase testing. We also have a couple of representative deals here from other small biotechs that had licensing events with a major pharmaceutical partner. So this is a very popular space in the last couple of years. Really since the FDA approval of the first siRNA by a company called Alnylam a few years back has really shown the light on the potential for siRNA. We have a great leadership team in place. Myself, I've got more than 20 years in cancer research and product development. Glenn Cazo has had a long career in the pharmaceutical industry, has participated in a number of different startups. He's had a leading role in 3 IPOs and raised over $500 million. Our financial strategist is also a life science scientist turned entrepreneur turned financial person, has raised more than $150 million for her companies. We've recently brought on 2 very high-powered advisors for clinical development and regulatory. Each have a long career in the pharmaceutical field. Participated in a number of FDA approvals. We have a great scientific advisory team in place. Narendra Vaish was one of the early members of the RNA inhibition fields. He was part of a company called Cerna Therapeutics that was bought by Merck in the early 2000s for more than $1 billion. We have a great drug development advisor who's been both in small biotech as well as J&J and others. We have a drug delivery advisor who's responsible for the discovery of this nanojacket technology. We also have the original discoverer of this family of PRDMs more than 20 years ago by the name of Shi Wang, who spent a career studying these PRDMs. We also have a great list of partnerships already in place and continuing to develop as we increase our capability for technology as well as testing. So we've partnered with folks to help us better deliver our siRNA, as well as test in vitro, in vivo, and ex vivo as well. We've had some good recognition and accomplishments over the last couple of years, including being a finalist at the Johnson & Johnson Innovation Quick Pitch, JP Morgan in 2020. We were named Buzz of Bio at the Bio International Conference. Also in 2020, we were part of a highly selective CLSI FAST program of California Life Sciences Institute, and proud to be a member of the Cube Accelerator program. We are raising money right now. We're doing a $15 million Series A. The purpose of this is to bring Arise 47 to the clinic with this IND enabling studies, as well as complete the human phase 1B studies in human patients to examine both efficacy and safety. A portion of this money will also go towards expanding the PRDM platform with the development of additional products for other cancer indications, including ovarian cancer, breast cancer, multiple myeloma, as well as this small molecule project to target PRDMs in a separate way as well. The outcome is a licensing event with a pharmaceutical partner. These $20 million plus type licensing deals, in addition to exit opportunities with an acquisition or IPO in the next few years. So in summary, precision medicine-based approach to target cancer at the root cause. We really think the way towards a cure for cancer is this precision medicine approach, really going after the true cause of the cancer. We have great multi-billion-dollar comparables in Epizyme, Constellation, and Alnylam for both this histone space as well as the siRNA space. We've made great progress, de-risked a lot of the development with positive in vitro and in vivo results. We have a high quality and experienced team in place, and we're also VC backed in our seed round by a company called Moneta Ventures. Has been great support for us in the past as well as moving forward. So I'll stop there. Thank you very much, and happy to take any questions.
Thanks, Brett. That's a really nice presentation. Are there any questions from the audience online or on site?
Yes, Michael, we have some questions in San Jose.
Yeah.
Thank you, Brad. Just wonder, it's a family of the proteins, and then maybe there's some conserved domains there. Is that possible that if you assume that each of the member is maybe necessary but not sufficient, that you can introduce loss of function for more than one members in order to have a more synergistic effect?
It is possible. We're doing some work to do the characterization of tumors to really see how much of that is truly going on. What we have done is some development work of combining various different siRNAs targeting multiple different PRDMs at the same time. We do see some small benefit from doing that, but finding the right model that has those disruption in multiple different PRDMs, we anticipate would have a larger effect with that combination therapy, which could be a possibility.
Yeah, and also the assumption that the expression profile for some cancer-related members are cancer-specific. In the cases not, would you actually, while formulating the nanoparticle, introducing some cancer-specific tropism?
Yeah, certainly. So there's a possibility for characterizing those tumors, and with that information, we can do a lot of different playing around with the siRNAs that are inside the drug delivery system, as well as targeting agents on the outside of the drug delivery system. We've done some work and are in the process of testing right now the addition of an aptamer on the outside of the drug delivery system to help selectivity to higher selectivity to cancer cells versus normal healthy cells.
Thanks. Is there any more questions from the audience? Right.
Hi, Brad. This is Tan Rasab.
Hey,
Great
Tan.
to see your pitch
Great
today.
to hear your voice.
Yes. One of the questions I wanted to ask is, which is almost actually promoting for my own company, is what do you see as the role of genetic sequencing and specifically tests, companion tests for products or medicines such as yours?
Yes, yeah, no, definitely. We clearly think there's a huge role for the companion diagnostic, especially when you've got a great biomarker, such as we do with the PRDMs, and other things as well. As you well know from your company, being able to select those patients based on their genetic markers makes all the difference in the world. You're already seeing it now with the targeted therapies, with the immunotherapies. It's really about the patient selection and finding the drug product that's going to work for that particular patient and their particular cancer. Definitely a crucial part of drug development these days, and we're really happy that you're working on that so we can combine efforts.
Thanks, Tan. Thanks, Brad. Due to the time, we probably have to move to the next company. Thanks, Brad, for the wonderful presentation. Have a good evening.
Thank you very much. You too.
The next company is going to be Inozi. So Inozi Life Sciences is a drug research and development company providing therapeutics for autoimmune diseases and cancer. The company was founded by a really famous scientist, Professor Mark Feldman, who discovered anti-TNF therapy, and also Dr. Michael Shepard, whose research is humanized antibody created the first monoclonal antibody therapy targeting cancer oncology encoded proteins. Today, Mike is going to talk about it. Mike is the CSO and president of the company. Michael, you can unmute yourself and share the screen.
There, can you see everything?
Yeah, you can try to share a screen now.
Oh, shoot, screen, okay. Is it shared?
No, it's not shared.
Oh, okay. I'm not sure what to do here.
Just click the green button down there, the share screen.
Oh, I got it. Thank you.
Yep, yep,
Yeah, I thought I was already there.
yep. And go full screen.
Okay. Is that better?
No, we cannot see the screen yet. Yeah, it's better.
No, that's good to hear.
Yeah.
All right. Thanks very much. Thank you for this wonderful opportunity to present to Cube. I'm sorry I'm not there in person. I was hoping I could be there in person. The presentations so far have been really fantastic. Now I think I'll tell you a little bit about Inosi and some of what we're going to say here will remind you of the earlier presentations, some aspects. What I'm really going to be talking about is one of the reasons why Inosi exists, which is to combine thinking about autoimmune disease and cancer. We can discuss more deeply how these two things are related, but just to begin, I'll let you know about who's in the company. I'm there. Dr. Feldman is a member. Jim Whitty, who's been a very famous venture capitalist, Pat Gray, and one of our longtime colleagues, Yasunori Kaneko. Our scientific advisory board has now 3 National Academy members and also one person, Alex Tarakovsky, who I'm sure will be a National Academy member soon. The 2 programs that I will talk about today are EN 1001, which is a specific TNFR1 antagonist. But I will be focusing on the growth factor trap technology, which we call Numodulin after the HER2 new receptor family. So autoimmune disease happens when the immune system attacks itself. I think probably most people in the audience and elsewhere know that already. Some of the interesting facts about it is there's no cure for it, and effective therapies are out there, but they have too much toxicity. They help, but not as much as you might want. Overall, the average is 3 women suffering as opposed to 1 man. But in some conditions, the ratio of women to men is as high as 10-to-1. The basis of our approach with TNFR1 has to do with problems that we know about, which are related to the TNF blockers. The TNF blockers are drugs like Humira, which bind TNF. When Humira binds TNF, it prevents TNF activating both of its receptors, TNFR1 and TNFR2. To oversimplify, which I apologize, but we need to do that for the sake of time, TNFR1 is the inflammatory receptor. So it's really the one you want to target. TNFR2 is naturally the counterbalance to the activation of TNFR1. So TNFR1 is actually anti-inflammatory. It has other roles too, which I've made a note here, and that is that TNF is critical to defend against opportunistic infections, most particularly tuberculosis, but a lot of other diseases too, which are normally controlled by the macrophage. Our ability to block TNFR1, but TNFR2 unlocks the door to a completely new way to treat autoimmune disease, which we can talk about now. Our early data with our TNFR1 antagonist is you can find in the two publications on the right. But the goal of it is shown on the left side of the slide. The TNF blockers knock down both receptors. Our drug only knocks down TNFR1. So that leads to preservation of regulatory T cells, which are the natural anti-inflammatory cells, and also leads to the preservation of our defenses against infection. The TNF blockers, or the inhibition of TNF inflammatory responses, has been very useful in the treatment of a number of diseases. However, because the TNF blockers inhibit both TNFR1 and TNFR2, thereby making patients more sensitive to infections, they are really not used for a lot of chronic diseases outside of autoimmune disease. One such disease is Alzheimer's. Another one is, as an example, the acute inflammation that occurs after a pneumotropic virus infects. So an example of a pneumotropic virus is COVID-19. And of course, now we know that COVID-19 is not going away, and COVID-20 isn't here yet, COVID-21, 22, and so forth. Even with therapeutics and prophylactics against COVID-19, a lot of people are still dying from it. The reason why is because when the virus goes into the lung, it immediately causes macrophages and neutrophils especially to invade the lung tissue and produce TNF as part of the innate immune response. That leads to inflammation of the lung, very much stiffening of the lung tissue, and inability to breathe. Those aspects of the disease do not become apparent until after the early stage. So there's an opportunity to prevent patients going on respirators if we could inhibit the TNF cascade. It cannot be done with TNF blockers because TNF blockers will make patients more likely for other infections. It can be done with a TNFR1 blocker. So there you have the background for our drug program for TNFR1 in the future of the treatment of autoimmune disease and other diseases. Next, I'll move on to a version of a growth factor trap. And a growth factor trap is a molecule that binds to a growth factor or a cytokine. And it's given a long half-life in serum because it's fused to some molecule like the FC region of an antibody or human serum albumin or something like that. In this case, the growth factor trap can circulate around, penetrate tumors, grab growth factors, and then go into the endosome where the growth factors are released, and the the growth factor trap can escape. Why would somebody make a molecule like this? The reason is that with, for instance, trastuzumab or Herceptin, what we see is on the average 50%. That's fantastic. As the inventor of Herceptin, I'm very happy with that, but not really, because 50% is only half full. So I walk around every day wondering about the other 50% and how to deal with that. Other growth factor inhibitors antibodies include cetuximab, otherwise known as Erbitux. Those don't do nearly as well as trastuzumab, really only helping less than 50% of patients achieve a marginal increase in survival, but not a very happy one, because the antibody, the receptors everywhere in the body. So that antibody binds to every tissue, and it is ADCC competent, so it can direct immune cells to kill whatever cells it binds to. It works, and it sells $1 billion a year, but I hope nobody in the audience ever has to take it. So what is it that limits the efficacy of these antibodies? In the last 20 years, a lot of work has been done on that topic. The answer is when we inhibit Herceptin, when we inhibit HER2 with Herceptin or the EGF receptor with Erbitux, what happens is the other receptors in that family increase their expression, and the tumor cells begin to make a lot more growth factor than they were making before. That's what leads to resistance, and that's why patients will often progress when they're being treated with these antibodies. One way to address this question would be to inhibit all of the important members of the EGF receptor family at the same time and achieve some synergy by doing so. Let's see. Okay, so this is what we are doing, what we've done. We take the extracellular domain of either EGF receptor or HER3, because HER2 doesn't bind a growth factor. HER2 is a funnel. So we take the extracellular domains of HER2 and HER3, and we fuse them with the FC domain, which actually comes from trastuzumab. And then we combine them together. So in our studies, I'll describe it a little bit more. Once we have made the homodimer EGF receptor antagonist and the homodimer HER3 antagonist, we can put those cDNAs into CHO cells, and they will make a family of 3 antagonists. They'll make the EGF receptor homodimer antagonist. They will make the HER1, HER3 heterodimer antagonist, and they will make the HER3 homodimer antagonist. We take that oligoclonal growth factor trap. It's like a mixture of antibodies, really. And we call that EN2642. That's just what we call it. In brief, you can just call it Numodulin. You don't have to call it EN2642. That's too hard to remember. So each preparation of the Numodulin contains a homodimer of EGF receptor, the heterodimer, and the homodimer of HER3. If we put that into culture with tumor cells in the presence of serum, and we compare it for the activity that we can see from pertuzumab or trastuzumab or cetuximab or even an anti-HER3 antibody, we can see that inhibiting all three of those receptors, HER1, HER2, and HER3, by basically starving them of growth factors, looks, at least in this experiment and in others, to be much superior to using antibodies together. So we can see that the pneumodulin, the combination of the three growth factor traps, has superior activity and is distinct from the approved drugs. An important aspect of any cancer drug, despite whatever activity it may have as a monotherapy, is to see that it has synergy with a commonly used drug like carboplatin. Similar to one of the previous talks, we've done the same kinds of experiments combining the pneumodulin with carboplatin to look and see on lung cancer cells what is the effect. We observed a strong combinatorial effect of the pneumodulins and carboplatin. We also see it if we use docetaxel, and we haven't looked at a lot of other drugs yet, but these two are very important because they're two of the most commonly used drugs, especially in lung cancer. So, sorry, here we go. Now, one of the things, the place where I can put autoimmune disease and cancer together, it's been known for quite a long time that in autoimmune disease, and particularly in rheumatoid arthritis, in the joint, there's a tumor-like structure that grows. It grows and it spreads inside the joint. It looks just like a tumor. Just like in cancer, a solid tumor is packed full of immune cells, and it's the immune cells that drive the growth and expansion of the cancer. In the pannus, which is the tissue inside of joints, the same thing occurs. The inflammation drives the proliferation of fibroblasts, which are then invaded by immune cells that produce inflammatory cytokines. If we can stop the growth of the pannus, we will stop the invasion of the immune cells, and we will stop the progression of the disease arthritis, rheumatoid arthritis. So that's what we did. We took a model called collagen-induced arthritis, which is a way of testing arthritis drugs. What you do is you immunize the mouse.
Hi, Mike.
Yeah.
Due to the time, we probably need to wrap it up really fast. So probably you still have 2 minutes to finish this. Thanks. Apologies.
2 minutes is plenty of time. Don't worry.
That's good.
So when you immunize the mice with collagen, they start to create antibodies that bind to the joints, and they develop arthritis. In order to prove that inhibiting the growth of the pannus will improve outcome in rheumatoid arthritis, we did the experiment shown here. Briefly, I just tell you what is the outcome. The oligoclonal growth factor trap inhibits the progression of arthritis in this model. Similar to the way etanercept can inhibit progression in this model. If you combine those two things together, the mice completely recover, and they look the same as non-arthritic mice. Those are the two panels on the bottom left of the slide. So the the illegal clonal growth factor cytokine trap can likely be applied to many different systems. And we'll be pursuing that in an OC along with our anti-TNFR1 program. Thanks for your patience.
Thanks, Mike. That's a really nice presentation. Are there any questions from the audience? I got a
Michael,
question.
you have a question online. We have a couple in San Jose. Go ahead.
You go first.
Okay.
Well, it's a privilege to listen to Mike's presentations, having attended to one of the previous presentations that he did last year, I think. It's always fascinating. Mike, I wanted to ask you, when do you think you're going to have this inhibitor for brain fog? Because I think with all this long COVID, which a lot of people are suffering, it may be an opportune time to have that sort of drug available much faster than perhaps you may have anticipated.
I think you're right. We are currently, raising money just like everybody else here, and our ability to raise money is pretty much what dictates the timeline. But we do have a lead molecule. We think it will be about 3 years to the clinic, unless, as you might guess, it would be possible to get there faster using a not quite approved form of the drug in patients suffering from a pneumotropic viral infection, a serious one.
Yeah, it's a really nice question, Tan. I think I can answer that a little bit as well. It's actually, there are a lot of companies that are doing the anti-inflammation as add-on for the vaccination as well, especially for Johnson & Johnson. So right now, Sanofi is actually putting a lot of effort on that, using the small molecule as a combination with the vaccination. So it's really, really interesting direction.
Thank you.
Yeah, hopefully I help you a little bit.
Yeah, thank you.
Hi, Dr. Shepard. I have a question that needs your maybe your perspective regarding the role R1 versus R2. There's several literature showing that it could be synergistic or antagonistic, or sometimes just sort of ambiguous in terms of relationship between pro-inflammation or anti-inflammation. Do you think the field actually have been kind of settled on such, research on the role of each of the two members?
There's not much discussion about whether or not TNFR1 is an inflammatory receptor. There is some discussion around TNFR2, and just like other things in immunology, the way that say regulatory T cells behave in a tumor microenvironment or in an environment like the pannus in rheumatoid arthritis depends a lot on the other cells that are there already. And our data suggests that in rheumatoid arthritis, in the animal model of rheumatoid arthritis, preserving regulatory T cell activity is extremely advantageous. We don't know what will happen in patients until we do it, but in the course of developing our thinking about this problem, we have a number of collaborators who are helping us to define the microenvironment, the molecular microenvironment that would be most favorable for our compound.
Yeah, I think maybe the key point is that you think your approach by targeting R1 will expand the therapeutic window. And that
Oh, absolutely.
Yeah.
Yeah. Unfortunately, it seems that that pharmaceutical companies have consistently underreported or understated frequency of serious infectious diseases and secondary autoimmune diseases that comes as a result of comes as a result of treatment with TNF blockers. So I think the big surprise is that these drugs, the TNF blockers, are 30 years old and nobody's made a significant improvement. Why not? I don't know why not, but I can certainly give it a try.
Yeah, sorry, just one last question. Would you envision that to dethrone Humira in the phase 3 setting, would you plan to do a head-to-head comparison against Humira to define that you actually had a much wider therapeutic index? And how much you're raising? Sorry, I actually forgot to ask.
We're raising $35 million.
Thank you so much.
Yeah, so head to head, I don't know if we'll have to do head to head because we actually haven't chosen the best indication yet. One possibility is once we've shown that the antagonist is safe, we could use it to treat early stage rheumatoid arthritis where patients are already in pain and the disease is progressing. But because of the issues with TNF blockers, they're not yet, physicians are not yet able to prescribe them because of the toxicities and side effects of the TNF blockers. So in that case, we would not have to go head to head, but happy to do it. I don't care. Go for it. That's what I say.
Mike, I can help you a little bit. The answer is you don't have to do that. For Humira, the mechanism is totally different from yours. Humira is doing the TNF, the NF-κB pathway, but you are actually targeting the TNF-R1 and R2. So the mechanism is totally different, and it's both anti-inflammation, but there's no competition between two of you guys.
Thank you. I appreciate all the help I can get.
Yeah, thanks. Due to the time, we're probably just going to finish this. Thanks a lot, Mike, for coming. It's a really wonderful presentation. Thanks a lot. Have a good evening.
You're welcome. It's been great. Thank you very much.
Thanks everyone for participating, all of the audience and investors in both the United States and China. And if you have any interest in any of the companies, feel free to contact us. I will surely connect you guys. You can always reach me by email. Can you put the info email in the chat box to make sure everyone knows how to contact us? We'll have more event in coming October. See you guys in October, and have a good evening. I'm gonna pass over the control to Brian on the floor. Probably
Michael,
you guys
thank
have
you.
a social later, right?
Absolutely. Wish you were here. Michael, thank you very much. Everybody in Hangzhou, good to see you. Thank you for joining us. Everybody here at Cubay in San Jose, thank you for coming. Great presentation. Cue the music. There's still more beer and wine. Before you go, we would like to take a small group photo up here if you guys are, would be so willing. So come on up. Again, thank you for coming tonight, and enjoy the beer.
And we see what we've become. In the cold light of day, we're a flame in the wind.